Anything? All right. So we'll go on and now discuss the immunoglobulin isotypes, allotypes, and idiotypes. Right. Now we're going to take a change of direction here. So far, we have been talking about the immunoglobulins as antibodies, and certainly that's what they are. That's their function. They function as antibodies. But an immunoglobulin is a protein, right? Basically, it's a protein. I mean, it does have some carbohydrate on there, but it's basically a protein. And so there's no reason why we can't use that protein as if it were an antigen. In other words, there's going to be some antigenic determinants on an immunoglobulin molecule that theoretically we can make antibodies against. So in this discussion of isotypes, allotypes, and idiotypes, we have to think of the immunoglobulin now in terms of the fact that it is an antigen that we're using to make antibodies against, and we're going to discuss the various types of antigenic determinants that you find on an immunoglobulin molecule. And the determinants that you find on the immunoglobulin molecules are called isotypic determinants, allotypic determinants, or idiotypic determinants. All right? And we're going to discuss each of those. So keep in mind we're dealing with the immunoglobulin as an antigen. Okay? And we'll start with the isotypes. So what are isotypes? Here's a nice dictionary definition. Antigenic determinants that characterize the classes and subclasses of heavy chains and types and subtypes of light chains. Okay? So what does that mean? Right? Now, if everybody would just close or certainly don't look at your laptops, all right? I want to just go through this little exercise, and if I see you looking at your laptop, you're the one I'm going to ask the questions to, okay? And you may know the answer to the question, but then I'm going to ask you why, okay? All right. Suppose we do this. Suppose we take some human IgM, and I inject that human IgM into a rabbit. Well, obviously, this is a protein, so this rabbit may recognize things on there that are going to be antigenic determinants, and the rabbit's going to respond to those and start making antibodies. So we can take the serum out of that rabbit, and we'll refer to that as an antiserum because it's a serum that contains antibodies against whatever we use to immunize with. So the question is, what will that antiserum react with? Do you think it will react with IgM, human IgM? Sure. I mean, that's what we immunized with, right? So it'll react with human IgM. Will it react with human IgG? That becomes a little bit more difficult, huh? Will it react with human IgG? Okay, we got a yes. We got a yes. Do we have everybody agree? It's a yes? Okay, we got to have a dissenter. Anybody else dissent? How many just don't know? Okay. Okay, we'll have yes, no, and abstain, right? All right. Somebody that said yes. Why did you say yes? Somebody want to volunteer?
Those two kind of go together in my mind.
Okay. Anybody else got an idea why? Besides just intuition. If they're variable regions.
If they both have the same specificity.
That's possible. But it's not likely that though you're going to have, remember when I take an IgM, I'm just taking a whole population of molecules. They have lots of different, specificities. So that's possible, but probably not likely.
Do you have any other things that would react with general antigen with foreign bodies? So would it develop?
I'm not sure what you're asking. I mean, we just simply took this IgM, injected it in, and the question is it going to react with IgG? Some people say yes. I told you yesterday when I said, what happens if, when we were talking about immunoglobulins and I said, this is IgG. What did I tell you? I told you that it's going to have everything there. So when I inject IgM, at the very least, I am also injecting kappa light chains and lambda 1 and lambda 2. Those are the same things that are present on IgG. So at the very least, when this animal recognizes this, anything on any anti-light chain activity that's present in this serum is going to react with IgG as well. This will react with all immunoglobulins, right? It'll react with everything. Because at the very least, there's the light chains in there stimulate the production of the antibodies, and those same light chains are on all these different classes of immunoglobulins. I told you. Okay? So it's going to react with everything. All right. Now, suppose I take this immunoglobulin, this antiserum, and I react it with IgG, IgA, IgD, and IgE. Right? And again, without me telling you how I can do this, but let's say that there is some way that after I let the antigen antibody complexes form, I can remove those things. So I can remove all the immune complexes that form with these. And then I have what is referred to as an absorbed antiserum. It's been absorbed. And so now the question is, what's this going to react with? Will it react with IgG? No, because now anything that could have reacted with IgG would have been reacted with this and has been removed. So all those antibodies to light chains and kappa light chains, all that's going to be removed. All right? So this is not going to react with IgG anymore. Will it react with any of these classes of immunoglobulins? Will it react with IgM? Yes, it's still going to react with IgM. Now it's going to react only with IgM. So this is now specific for IgM. What's it reacting with? What do you think it's reacting with? What makes IgM IgM? The heavy chain constant region. So any determinants that are present in the heavy chain constant region that are unique to the mu heavy chain is going to be what this is reacting with. So this is reacting with determinants that are specific for the mu heavy chain. Those determinants that are unique to that heavy chain are referred to as isotypes. Now I can go through the same process. I can immunize with IgG here and come down here and then absorb, change this to M, and I could produce an antiserum that is specific for G. All right, the gamma heavy chain, or the gamma 1 heavy chain if I want to do it that way, or gamma 2. So each class and subclass of immunoglobulin has its own unique set of antigenic determinants that are specific for that particular heavy chain. Those are the isotypes. And I can do this same exercise with light chains. I can inject kappa light chains up here, absorb with lambda light chains, and I could produce an antiserum that recognizes specific determinants that are only found on kappa light chains, or only found on lambda light chains, or lambda 1, lambda 2, lambda 3. So each class and subclass of immunoglobulin has its own unique set of antigenic determinants that are unique to that class or subclass of heavy chain, type or subtype of light chain. And those are called isotypic determinants, or the isotypes. All right. Is that clear what they are? All right. So where are they located? Well, by definition, what makes a mu chain is the heavy chain, right? So the isotypic determinants are going to be localized to the constant region of the heavy chain, because that's what makes it a mu. In the case of the light chain isotypes, it is located on the constant region of the light chain, because that's what determines the type of light chain, is the constant region. All right? So they localize to the constant region of the heavy chain or the constant region of the light chain. Now you'll notice that in this particular diagram, I put in this part here is, of course, corresponds to CH1, right? This is the first domain of the constant region. You notice I put it in gray. And the reason for that is, I mean, theoretically, there are isotypic determinants in this portion of the molecule as well. In practice, when you do the type of immunization that I just described, where you inject the whole molecule, typically you don't produce antibodies against that part of the molecule. The antibodies are against this part of the molecule or against this part of the light chain. So, I mean, theoretically, they should be there, but to get them, to identify those isotypes, you have to do things a little bit differently than just the way we described it. All right, so let's take a look at the situation. We have an IG1 kappa and an IgM kappa. All right, these two immunoglobulins will have different heavy chain isotypes, right? Because this is a gamma 1, this is a mu, so they have different heavy chain isotypes, but they have the same light chain isotypes, right? Because they both have kappa chains. All right, so where do we find the isotypes? What are the occurrence of these? The isotypes are found in all immunologically normal individuals. Okay, so every one of us, if we don't have some sort of immunodeficiency disease, every one of us expresses all of the isotypes. We all have IgM, so we have all the IgM isotypes. We all have IgG1, IgG2, we all have IgA, we have all the classes. Similarly, we all have kappa and lambda chains, so every one of us expresses all of these isotypes. Now, in fact, that's what the term iso means. The term iso comes from the Greek meaning the same among members of the species. So every one of us has all of these isotypes if we're immunologically normal. All right. So what are they used for? Well, the isotypes are what is used to identify and measure immunoglobulin levels. When you go and you're going to send a patient sample down and you want an immunoglobulin level down, they're not going to go and isolate the immunoglobulin and sequence it to try to determine what constant regions the immunoglobulin has. I mean, that is a huge task. So that's not what a clinical lab does. What the lab is going to do is they're going to go over to the shelf, and there's a series of reagents. This antibody will be specific for IgM. This antibody will be specific for IgG1. And they use those reagents then to evaluate the immunoglobulin levels of each particular class or subclass of immunoglobulin that you may request that you want to get information on. So the isotypes are what the clinical labs use, basically, to measure immunoglobulin levels. And when we talk about antibody reactions, and we'll go over some of the tests that are used in some of the labs for doing these kind of things. These things can also be helpful. Detecting isotypes can also be helpful in helping diagnose B-cell tumors. Remember I said B cells have on their surface immunoglobulin. So they have certain isotypes expressed on that immunoglobulin that's on the surface of the cell. Now, if we look in humans, if you look at B cells and you ask what proportion of B cells express kappa chains and what proportion of the B cells express lambda chains, it's roughly 50-50. It's different in other species, but in humans it's roughly 50-50. So half of our B cells, if I were to take a sample of your cells and look at them, whether they have kappa or lambda, half of them will have kappa, half of them will have lambda. Now, if you have a B-cell tumor, though, you get a malignant proliferation of a clone of cells. So one clone of cells starts proliferating, and you have a lot of B cells then, all of which are derived from one clone. So they will have just either a kappa or a lambda, right, on them. So by looking and counting the number of B cells that express kappa and lambda isotypes, if you find in your patient that there's a preponderance of kappa, you know that there's something wrong. It's supposed to be roughly 50-50. So if 90% of your cells are expressing kappa, that's an indication that you're dealing with a B-cell tumor that's expressing a kappa light chain on its surface. All right. So this can be helpful in looking at B-cell tumors. Immunodeficiencies. I mean, this is how you do. One of the ways that you help diagnose the immunodeficiencies. What's going to happen if a person has an IgA deficiency? He's not going to have any IgA in his serum. You take some of his serum and you measure the amount of IgA that's in that serum, and you'll see that it's lower than the normal range. Okay, so these isotypes are what are commonly used in the labs, basically, to identify all these different classes and subclasses of immunoglobulins in the types and subtypes of light chains. So they're very important, actually. They're used a lot. All right. Any questions on isotypes? All right. Then let's go on to allotypes. And again, here's our nice dictionary definition. They're antigenic determinants specified by allelic forms of the immunoglobulin genes. All right, so what does that mean? I like to do this one by analogy. Every one of us in this room has hemoglobin. I hope. Okay, so we all have hemoglobin. But it doesn't mean that every one of us has exactly the same hemoglobin. Most of us probably have hemoglobin A, which is the hemoglobin associated with adult hemoglobin. But there may be some in this class that have hemoglobin S, the hemoglobin associated with sickle cell anemia, sickle cell trait. Do you know what the difference is between normal normal hemoglobin A and hemoglobin S? 1 amino acid difference, all right, in I think it's the beta chain of hemoglobin. So it's just 1 amino acid substitution, okay? So just as in the case of hemoglobin, where you can have everybody having hemoglobin but being slight differences, the same thing applies in the immunoglobulin molecules. Every one of us has got IgG. But that doesn't mean that your IgG is exactly the same as my IgG. I may have inherited a gene that specifies a particular amino acid at this position in the chain. You may have inherited a gene for the immunoglobulin that specifies a different amino acid. And therefore, if I were to take my immunoglobulin and inject it into you, and take your immunoglobulin, or IgG, and inject it into me, the possibility exists that I would respond and recognize your immunoglobulin as being different. That 1 amino acid difference may have created a new epitope that I would recognize as foreign. I would make antibodies against it. You could make antibodies against my immunoglobulin. All right? Those differences are the allotypes. Okay? So differences that are specified by different allelic forms of the immunoglobulin genes can give rise to these allotypes. All right, so where do we get and how do we identify the allotypes? Obviously, the only way that you know an allotype exists is if you have an antibody to detect the allotype, all right? Now, we don't normally go about taking my immunoglobulin and injecting it into you or your immunoglobulin and injecting it into me. We don't normally do those things, all right? But there are instances where, in fact, we do that. And one of the places is in blood transfusions. If you've ever had a blood transfusion, they will match you for blood group antigens, but they don't match for immunoglobulin allotypes to make sure that you have the same immunoglobulin allotype. And the reason that it's not matched for immunoglobulin allotypes is that clinically it doesn't make any difference if you inject a person with a different allotype immunoglobulin. They'll just make antibodies against it, okay? It's not a problem clinically, so we don't do it. So anybody that's received a blood transfusion potentially is a source of antibodies that would be directed against the allotype of whatever the donor had, right? The donor allotypes. Another place that we get these antibodies is from women that after they've given birth. At birth, there's a rupturing of the membranes, and there's a mixing of fetal and maternal circulation, all right? And so what happens? If the child has inherited an allotype from the father that the mother doesn't have, the mother will essentially become immunized against that allotype. So women then become a source of these anti-allotypic antibodies that are used to detect the allotypes. All right, so where are they found? Theoretically, there's no reason why the allotypes couldn't be anywhere on the immunoglobulin molecule. In theory, they should be everywhere. But in fact, all the known human allotypes are located to either the constant region of the heavy chain or the constant region of the light chain. Again, there's no reason why that has to be the case. In fact, for example, in rabbits, there are variable chain allotypes in rabbits. So, I mean, it's possible. But for some reason, at least all the known ones in humans, that doesn't mean that there aren't some unknown ones, but all the known allotypes in humans are located to the constant region of the heavy or the constant region of the light. So let's look at a situation. We have here two IgG1 kappas from two different people. Okay? These are both IgG1 kappas, so they share the gamma 1 isotypes, which are indicated in yellow here. You have the same gamma 1 isotypes. You have the same kappa isotypes because the isotypes are the same. But they have different allotypes shown in orange because they're from different people. This one has one set of allotypes. This one has a different set of allotypes. On both the heavy chain and on the light chain, you can have the allotypic differences as well. Okay? So the allotypes are located then in these constant regions. So where do they occur? Well, obviously, each of us has his or her own unique set of allotypes. All right? Now, some of us may share certain allotypes, that's true, but whatever genes you've inherited will determine what allotypes you have. Whatever genes I've inherited determine my allotypes. Okay? So the term allo comes from the Greek, meaning different among members of this species. So there are differences. Each of us may have some differences from each other. Now, actually, let's take a break now, and then I don't want to introduce the usefulness of these allotypes come from an understanding of the genetics, and that's what we'll cover next. So let's take our break now, and then we'll pick up here. Okay, can we get started? Is it okay? All right. As I said, the usefulness of the allotypes and what their value is to us is really comes from an understanding of the genetics of the allotypes. So what I want to do is I want to just talk to you a little bit about some of the human allotypes, human immunoglobulin allotypes, and then about their genetics and then how they're used. All right, first of all, just as far as the nomenclature is concerned, the nomenclature really gives you everything that you need to know about the particular allotype. All the immunoglobulins the allotypes are indicated with the nomenclature like this. The first part of the name tells you what chain it's on. So obviously this is on the gamma 1 chain. So this is going to be an allotype on the gamma 1 chain. The M is the symbol that this is an allotypic marker. So this is an allotypic marker on the gamma 1 chain. All right? And then following that, in parentheses, is some number, and that tells you which allotype you're dealing with. So this is allotype 3 on gamma 1 heavy chains. This is allotype 1 on kappa light chains. All right? So the name will tell you what chain it's on, and it'll tell you which allotype it is. And here's an example of some allotypes. So for example, by the way, you may see in some older texts, instead of a number in the parentheses, they'll have a letter. That is an older nomenclature, and if you see it, don't get concerned about it. It just they use letters instead of numbers. So the allotype 3 is actually in the old nomenclature of allotype F. All right, but what allotype 3 is, G1M3 is actually an arginine at position 214 in the chain. So if you have a gene that specifies arginine at position 214, then you will express allotype 3. You will be G1M3. All right. In contrast, if you have a gene that specifies lysine at that position, then you will be G1M17. All right. So you will have that allotype. Some of the allotypes are a little bit more complex. It's more than a single amino acid. G1M1 is actually this sequence of amino acids, arginine, asparagine, glutamate, leucine, at these positions, okay? So if you have a gene that specifies those amino acids at that position, then you will express that particular allotype. And you have allotypes on the light chains, like the KM allotypes. The KM1 is valine leucine at those positions. And KM3 is alanine valine at those same positions, all right? So depending upon what gene you've inherited, that will determine which allotypes that you have. Now, the genetics of these allotypes like the KM allotypes and the G1M allotypes like 3 and 17 and KM1 and KM3, see, they represent alternate amino acids at the same positions in the chain, all right? Those types of allotypes are inherited as codominant autosomal genes, so that neither one is dominant over the other. They both are expressed, so that if you are an individual that is a heterozygote, KM1, KM3, for example, heterozygote, you will be, if I were to take some of your immunoglobulin and look for the presence of the allotypes, I would find both 1 and 3 expressed, all right? And they follow just simple Mendelian genetics. If we have a heterozygote crossed with a homozygote KM1, roughly half of the offspring will be KM1, and the other half will be heterozygote. Okay, so it's simple Mendelian genetics. Now, although these are expressed as codominant alleles, so that in a heterozygote, both the KM1 and the KM3 are expressed, if you look at any individual and immunoglobulin molecule, all right, so now just look at one molecule. Each molecule will be either KM1 or KM3, but never both. Because remember, an immunoglobulin molecule, they're composed of 2 identical light chains. There's no differences between them. All right, so in any individual molecule, we'll have either KM1 or KM3, but never both. But in the population of molecules, some will be KM1, some will be KM3. Now, the reason that happens, that individual molecules only express one or the other, is that there is a phenomenon called allelic exclusions. And what that means is that B cells exclude one allele. When they express the immunoglobulin genes, they exclude one of the alleles. Remember, we have 2 alleles for every, well, 2 alleles for all of these genes, because we've inherited one from our mother, one from our father. All right? So the B cells exclude one. It's called allelic exclusion. And therefore, the B cell only makes, either are the expresses the gene from the mother or the gene from the father, never both. And that's why the immunoglobulin secreted by that cell only has identical light chains and expresses only one light type, or one kappa light chain allotype. All right? Is that clear? Yes.
Going back to the million, it'll be about 50.
They're roughly 50-50. Right. So about half the molecules will be KM1, half the molecules will be KM3. All right. All right. Now, it is possible, however, to have more than one allotype on the same chain. So, for example, if you have a chain like, if you have an IgG1 like this, at position 214, if you express lysine, and at positions 355 to 358, you express this set of 4 amino acids, then obviously that particular chain will have 2 allotypes. It'll have 17 on it and 1 on it. Okay? So it is possible in these situations where they're not, where the allotype does not represent different amino acids at the exact same position, then it is possible to have 2 allotypes on the same chain. But it would never be possible to have an arginine and a lysine at the same position in an individual chain, right? You have to have one or the other. So in these cases, you do get on one chain you may get 2 immunoglobulin allotypes expressed. But in the case of the KMs or GM1, or GM3 and GM17, that could never happen. You'd have to have one molecule could express 3, one molecule could express 17, but you could never have a molecule that expresses both in those instances. All right. So what are the allotypes used for? Well, there are a number of practical applications of the use of allotypes. One is in monitoring bone marrow grafts. What do you do in a bone marrow graft? In the recipients, for whatever reason, the bone marrow, if it's either not functional or has been destroyed because you're trying to deal with a tumor, you get rid of the recipient's bone marrow, basically, by irradiation or by chemical treatment. And you're going to repopulate the bone marrow with the transplant from the donor. Now, again, they will match the donor and the recipient for all sorts of things, all right? For blood group antigens, they're matched for their blood, they're matched for tissue type, and then so on, as close as they can get to match. And then they do the transplant. But they don't match to see whether or not you have the same immunoglobulin allotypes. And the reason is, again, because clinically it doesn't make any difference. There's no problems clinically if you receive a bone marrow graft from a bone donor with a different allotype. It doesn't make any difference. So it's not matched. But we can make use of that then in finding out whether or not that bone marrow takes. If the bone marrow is taking, what's going to happen? The bone marrow is going to repopulate the bone marrow of the recipient. Those cells, the stem cells in that bone marrow are going to begin to differentiate into lymphoid cells, and some of those lymphoid cells are going to be B cells, and some of those B cells then are going to start secreting antibody. But now they're going to be secreting antibody of the donor allotype. So by typing the donor and the recipient for the allotypes, even though you don't necessarily have to match them for that, you can then, know whether or not the bone marrow graft is taking if you can start detecting donor allotypes being expressed in the patient's serum. You know that the bone marrow graft took, and it's actually the cells have differentiated into B cells that are secreting antibody. Now, obviously, that's not the only thing you do in monitoring these patients, but it's one of the things that is used to see whether or not the grafts are taking, is by looking to see if the donor allotypes are being expressed in the recipient. Another place it's used is in forensic medicine. The allotypes can actually be detected in semen, in dried blood, in saliva, and so there's application in the forensic field for using allotypes to help identify people. And not so much nowadays anymore, because now, of course, we've got the DNA technology where we can identify, whether or not a person is the father of a child. But in paternity testing, it had been used a lot, and to some extent it is still used, because by these tests you can't prove, when you're looking at allotypes, you can't prove paternity, you can only disprove it. All right? You can never prove that a guy is the father. You can disprove a guy is. So, and you can use the allotypes, and again, from the use of the genetics. If you had a situation where, let's say that the child is KM1, KM1, or I'm sorry, is a heterozygote. Let's say it's KM1, KM3, and the mother is KM1, KM1. If the guy, the putative father, is KM1, KM1, you know that that can't be the father, all right? So in paternity testing, then you can use these allotypes, at least to disprove paternity. Nowadays, of course, the DNA technology can, with a high degree of certainty, say that this is the father, so this stuff is certainly being replaced. But there's still some application of the allotypes in that field as well. All right. Any questions about isotypes or allotypes? And I don't want to go on because those are a little bit easier. This is the difficult one. Okay? So if isotypes and allotypes are not clear, now's the time to ask the questions. I mean, obviously you can come later too, but if you've got them, let's deal with them now before we go on.
When you're talking about the cell and how they can only express one allele, that is per individual B cell. Like you can have like one B cell with this one.
Exactly, and that's why in the population of molecules, some of them have one and some of them have three, KM1, KM3, because some B cells excluded the 3 allele, another B cell will exclude the 1 allele, and so you get both different immunoglobulins in the population of molecules. Absolutely. Any other questions? Yes? Is it? Oh no, it's kind of. I mean, if we were to look on this here, we'd see a bunch of these different. I mean, that table I gave you, it was just a small number. I mean, there's a whole slew of these allotypes, okay? And I can guarantee you, we probably would not find two people that have exactly the same allotypes in this room if you looked at all of them. Now that we share a lot of them, obviously, but if you looked at the whole group, it would, you probably would never find anybody with exactly the same allotypes. No. These are all normal situations. Okay? Anything else? Yes?
When you were talking about like the nomenclature of the allotypes,
Right?
when it like takes essentially one, what did you say the number system?
It just specified which allotype you're talking about. That's allotype 1. KM1 would be the allotype 1 on the kappa light chains. It's just,
The order they found them?
yeah, it's the order they found them usually is what it goes. Yeah, that was probably the first allotype found on the kappa light chain and so on. Okay? All right. Idiotypes. Again, our definition. Unique antigenic determinants present on individual antibody molecules or molecules of identical specificity. So what does it mean? Again, I want to do this one by a little experiment. So close your laptops. All right. Suppose I were to take a haptene carrier conjugate, DMP-BSA. DMP is the haptene. It's dinitrophenol, just a small molecule, okay? The carrier is BSA. It's bovine serum albumin. It's just a big protein, an immunogenic protein. And I make a haptene carrier conjugate. So I chemically coat the DMP to the BSA. And I inject this into a mouse of a strain A. Now, I need to tell you a little bit about mice strain. Laboratory mice have been inbred by brother and sister mating for 20, 30, 40 generations. And so they're highly, highly inbred strains of mice, okay? And so they've been inbred so much that essentially every mouse is genetically identical. They're all identical twins for all practical purposes, so genetically, okay? So we're going to take one of these strains of mouse, so we'll call it mouse strain A, and we're going to inject our haptene carrier conjugate into the mouse. Well, the mouse is obviously going to respond, right? And the mouse will make antibodies against the BSA, the carrier determinants, But in addition, it's going to make antibodies against the haptenic determinants, right? And those antibodies against the haptenic determinants will react with the free haptene, right? That's what we said earlier. So we have in this population of molecules, some of those are going to be anti-DMP antibodies. In other words, antibodies that will react with the free haptene. Some of them will be. All right. Now, again, if you would give me that it's possible for me to do this without me telling you how at this point, suppose it were possible for me to isolate those anti-DMP antibodies so that I could pull out those antibodies and only the anti-DMP antibodies, all right, and get rid of all the rest of them. Now I have got purified anti-DMP antibodies. And I inject those antibodies into a strain A mouse. Not the same mouse up here, a different individual, but of the same strain. Okay? What will I get? Would you expect this mouse to make antibodies to any isotypic determinants that are present on those anti-DMP antibodies? Would you expect the mouse to make antibodies to any of the isotypic determinants? No. Why? There are no differences. I remember isotypes are present on all members of the species. So you're not going to get any antibodies against the isotypes. Would you expect them to produce any antibodies against any allotypic determinants on those? Why? No. They're genetically identical. There are no allotypic differences. Okay? So you won't get any anti-allotype. You won't get any anti-isotypic antibodies. Is there anything special about these antibodies that this mouse might be able to make antibodies against? The idiotype, right.
Their antibody, the variable would be specific for something that they've never encountered before, so they wouldn't have any.
Exactly. The combining site of this antibody that allows it to bind DMP is very, very unique, right? It's a particular set of hypervariable regions that create the combining site. So the combining site of this antibody is different. This mouse would see that combining site and make antibodies against the combining site of the anti-DMP antibodies. Well, what makes up the combining site? The hypervariable regions, right? So it's the antigenic determinants that are created by the hypervariable regions of the immunoglobulins that are called the idiotypes. Okay? All right, does that make sense? When any antibody that, in this case, has specificity against DMP, has a very unique set of hypervariable regions that allows it to make a combining site that will bind DMP. And so those hypervariable regions then can be an antigenic determinant that this mouse recognizes and starts making antibodies against. So this is antibodies against the binding site. And more specifically, they're antibodies against the hypervariable regions. Now, the actual determinant, the antigenic determinant, although it's certainly influenced by the hypervariable regions, may extend somewhat into the framework regions as well, but it's really dictated by those hypervariable regions. And, of course, the hypervariable regions are found on both the heavy and the light chains. And so there can be idiotypic determinants on both heavy chains and light chains because they both have hypervariable regions. Okay? Some of you are looking kind of shell-shocked. Yes?
So the first mouse that produced that antibody doesn't make the antibody against the antibody?
I'll come to that in a second. Good question. I'll come to that in a second. All right. Is it clear what the idiotypes are? All right. So if these are determinants that are created by the hypervariable regions. Now, as I said, you can find them on both the heavy chain and the light chain. Sometimes, however, the actual determinant becomes a conformational determinant that is created by both the hypervariable region of the light chain and the heavy chain. So in other words, it's some conformation that involves both hypervariable of the light and hypervariable of the heavy. In those instances, obviously, if you separate the heavy and light chain, you destroy the epitope. So it's if a conformational determinant, you destroy the epitope. But in other instances, it's directed only against say, the variable region of the heavy chain or only against the variable region of the light chain. In that case, you would not destroy the epitope by separating the chains. So you will find both conformational and sequence determinants in these as well. Okay? All right, is it clear what the idiotype is? It's an antigenic determinant that's created by the hypervariable region. Every antibody, then, of different specificity has a different idiotype. Because the specificity is determined by the hypervariable regions, so every different specificity represents a different idiotype. All right? Okay, so where are they going to be located on the immunoglobulin molecule? Well, obviously, they're going to have to be in the variable region, right? Because that's where the hypervariable regions are found. They're going to be up in the variable region of the heavy chain or the variable region of the light chain is where you're going to find the idiotypic determinants. So let's look at a situation here. We have two IgG1 kappas from the same person now. But one of them is against antigen A, and another one is against antigen B. These immunoglobulins will have the same isotypes indicated in yellow, right, on both the heavy chain and the light chain. They will have the same allotypes. They're from the same person on the heavy chain, the same allotypes on the light chain. But they will have different idiotypes, okay? This has one set of idiotypes. This has a different set of idiotypes because they have different combining sites. So every antibody with the different specificity has a different idiotype. Okay, so what are they used for? What's the usefulness of the allotypes? Well, immunologists like them because now we finally have a marker for the variable region. All the other things were for the constant region, but that's not important for you. One place where it is important is that they're involved in regulating immune responses. And let me illustrate what I mean by that. And this speaks to your question, okay? When I take an antigen and then immunize somebody with an antigen, it leads to the production of antibody. But for our purposes, I'm going to call this idiotype, right? Because the antibodies have a particular idiotype. So let's call it idiotype. So we're going to get the production of some idiotype, okay? Because the antibodies will have a particular combining site. All right. Now, once this is made, and this is the question that you had, once this is made, that's something that we've never seen before. So what happens is that the presence of this leads to the production of another antibody directed against the idiotype. In other words, an anti-idiotype. Now, it turns out that if you look at these, if you analyze these anti-idiotypic antibodies, they actually fall into two types. There are some of them that will actually stimulate the production of more idiotype, and some that will inhibit the production of idiotype. So what really controls then the amount of idiotype antibody that we make is three things. It's the amount of antigen that's there driving this production of this, as well as the ratio of the positively regulating and negatively regulating anti-idiotypes. So what controls this is all three of these. Is antigen there driving it this way? And then do we have anti-idiotype positively and negatively? What's the ratio of these two that regulate this? So the concentration of this is regulated by antigen as well as anti-idiotype. Okay? But this is just an antibody, right? So it's got an idiotype. So that leads to the production of an anti-idiotype, which can be positively or negatively regulating. So what regulates the level of the anti-idiotype is the presence of the idiotype as well as the presence of the anti-idiotype. Then positively and negatively regulating, right? But that's an antibody. And it's got an idiotype. which leads to the production of an anti-idiotype, and so on. So you get the picture. So you have this network of idiotype, anti-idiotypes interactions that help regulate these immune responses and monitor and keep the levels of these antibodies present, okay? So this is referred to as the regulation by the idiotype, anti-idiotype network. And in fact, you can actually, it's been demonstrated experimentally. For example, if you inject an animal with an antigen, you obviously can detect the idiotype, which is the antibody. You can actually detect the anti-idiotype, the anti-anti-idiotype, and the anti-anti-anti-idiotype. They've gone back experimentally and shown 3 levels back that you can actually detect these different antibodies actually being made in the individual. Yes? Well, obviously, if you don't want to just keep making antibody, if you just kept making antibody all the time, it would be no use. Once the antigen is gone, you don't need it anymore. So you want to control it then. So this just helps control the level. So what drives it is, as long as antigen is present, we're getting it. Afterwards, we can maintain some level of the thing by these as well. Okay? Yeah. No, in fact, the reason I didn't want to confuse, this gets to be kind of hard to conceptualize. So I put it in a separate mouse just to make it a little bit easier. But no, exactly the same thing would have happened in the first mouse that got injected. Okay. Yes, the anti-idiotype is something against the hypervariable region of this antibody molecule. And this one is an antibody against the variable region combining site of this antibody. Okay? Okay? No, it has got a combining site against it is an antibody against the combining site present on these antibodies. And this has an antibody against this one. Okay? Say that again? Yes, right. This is the only thing that will bind to this. Right. This is the only thing that will bind with antigen. Okay? All right. Another place where idiotypes, there's a lot of interest in idiotypes, is in the vaccine area, to try to develop what I refer to as anti-idiotype vaccines. So how can you use an anti-idiotype as a vaccine? Well, let's go back to this. We saw what happened when we injected antigen. We get this production. We get these positively and negatively regulating anti-idiotypes. If it were possible to selectively pull out these anti-idiotypes that positively regulated the idiotype, okay, if you could do that, and that is obviously the big trick in this. If you could do that, then to produce this antibody, you wouldn't need antigen anymore. That's all you'd need is the anti-idiotype. You can inject the anti-idiotype, and you would get the production of the antibody. So, I mean, why would you want to do this? Why not just inject antigen? Well, if the antigen is particularly toxic or dangerous to administer, and you don't want to do this, this is an alternative approach, okay, to inject the anti-idiotype. So there's a lot of interest in trying to develop these anti-idiotype vaccines as a way of getting around the fact that we can't always inject the antigen because it may be too toxic. Okay? Another place where idiotypes and anti-idiotypes are used is in the treatment of B-cell tumors. Remember we said B cells have on their surface immunoglobulin, IgM and IgD. They have the same specificity, so they have a particular set of hypervariable regions. That means that those B cells, every B cell expresses a particular idiotype, depending upon whatever the combining site of those antibodies are. Now, what happens when you have a B-cell tumor? You get a malignant proliferation of a clone of cells. So that clone of cells starts dividing, and you have lots of these cells, right? And then you have a malignant proliferation of these cells. So all the cells in the tumor that are tumor cells will be expressing the same idiotype. So if you could produce an antibody, an anti-idiotype, to that idiotype that's present on the tumor cell, then you can treat the patient with that. You can administer the anti-idiotype. The anti-idiotype will very specifically bind only to the tumor B cells, sparing all the normal B cells. It's highly targeted to that, and it can kill the tumors. It can kill the tumors maybe because it fixes, the antibody fixes complement and relies on that, or what's more typically done is it's possible to couple to the antibody molecules toxic materials that then kill the cell. So you use the anti-idiotype to target specifically the tumors, and then you kill only the tumor cells, sparing all the normal cells. Yes? And that would happen. That would certainly happen. Or it could happen. All right. But you treat and if you kill off all the tumor cells, you've treated the patient at least, as long as you can get the cell. Yeah, obviously it's going to take some time for that to happen. So you've got some time that you can work, but ultimately that theoretically can happen.
Like each person that has a B-cell leukemia, it's like you have to, as a physician, have to like take part of that and figure out what the idiotype is.
Yeah, actually what they do, you have to take out some of the tumor cells, get the idiotype, and then you prepare the anti-idiotype from that, and then infuse it back into the patient. So yeah, obviously it's tailored therapy, because every tumor is going to have its own idiotype. Okay? Now, we haven't talked a lot about T cells, but I will tell you that the same thing applies to T cells, because T cells also have a receptor on their, for antigen on their surface, and those receptors also express an idiotype, because they have hypervariable regions as well. And so they express an idiotype. And so, T cells have idiotypes as well. So there's no reason that you couldn't produce an antibody, anti-idiotype against the T cell idiotypes and use those for the treatment of T cell tumors as well. Yes?
This is still in research?
Yeah, pretty much so. But it's clearly, the direction that people are heading. They're trying to do these kind of things.
Have one noticed that there's like with among like the population that happens to cause tumors?
Not that I'm aware of. You're looking for a general reagent that could be used and then to treat. Not that I'm aware of. All right. Any questions on allotypes, idiotypes, or isotypes?